Hi everyone, my name is Dr. Sinead Blaber and I'm the Director of Clinical Development and Medical Affairs at Regeneus. I'd firstly like to thank the organisers of the Cell and Gene meeting on the Mesa for giving us this opportunity to talk about our company Regeneus and our two product platforms, Progenza and Cygnus. This is our forward-looking statement for your reference. So Regeneus is a Sydney-based biotechnology company that was founded in 2007 and listed on the Australian Stock Exchange in 2013. We're led by an experienced senior management team that you can see there on the right of your screen. And we have over 80 patents worldwide that underpin our two platform technologies, Progenza and Cygnus. So I'll be going through both of these today. Progenza is an allogeneic adult adipose-derived mesenchymal stem cell product. But our point of differentiation is that we incorporate the bioactive secretome along with our mesenchymal stem cells. Our Cygnus product is a cell-free product that is really harnessing the power of the bioactive secretome. For Progenza, we signed a license and collaboration agreement with Kyocera. So they have the rights for Progenza for knee osteoarthritis in Japan only. But we're seeking strategic partners for development and commercialization of Progenza for knee osteoarthritis in other major geographies ex-Japan. For Cygnus, we're also seeking global partners for a variety of different indications. This is really just to give you an overview of our development pipeline. We're targeting phase 2 trials of Progenza for knee osteoarthritis in both Japan and the US next year. And for Cygnus, towards the end of this year, beginning of next year, we'll be running a phase 1 trial of our Cygnus product. So now just to take you through what Progenza is and the data we have. Progenza is an allogeneic adipose-derived mesenchymal stem cell product, but as I mentioned, we harness the power of the bioactive secretome. So we cryopreserve our MSCs with the bioactive secretome that's comprised of cytokines, chemokines, growth factors, extracellular vesicles, all those sorts of good factors. And we do this for two reasons. One, because it improves the viability and the functionality of our MSCs, meaning we can have a truly off-the-shelf product. And secondly, because that bioactive secretome has a therapeutic benefit in its own right. Some other features of our product are it is an adipose-derived MSC cell line, so there's 500 to 1,000 times more MSCs in adipose tissue than bone marrow. And they've also been shown to be more immunomodulatory. And we also don't do any genetic modification or reprogramming of our cells, making them safer. In terms of the mechanism of action, it's well known now that mesenchymal stem cells don't exert their main therapeutic benefit through their differentiation capabilities. Instead, we focus on the immunomodulatory capabilities of our Progenza product, and really the ability of the product to switch the environment in an osteoarthritic joint from one that's very pro-inflammatory to an anti-inflammatory environment that's more conducive with repair and regeneration by the endogenous cells that are there. This is just to give you an overview of our CMC process. So we undertake a very rigorous screening of our donors to strict GCP and proprietary criteria to make sure we've got the best cells to go forward with our Progenza product. We then isolate the MSCs and expand them in vitro to produce millions of doses of our MSC product that's been cryopreserved with the bioactive secretome. This sort of large-scale approach to manufacturing means we can have a lower cost of goods compared to other cell therapies out there, such as autologous or pluripotent stem cell therapies. We also have really well-characterized final product and established quality control and product release criteria. And our final product will be available for our phase 2 trials in both Japan and the US next year. So to delve into some of the results we have for our Progenza product, we've conducted single-dose acute toxicology studies in animals for Progenza, and we've established safety both at the intended human dose and at doses higher. But really encouragingly for the Regeneus team, we saw evidence of disease modification in a validated surgical osteoarthritis model. So we utilized a lateral partial meniscectomy model in rabbits and demonstrated that Progenza was able to halt disease progression in the weight-bearing zone of these animals. So this green bar here was statistically significant for the vehicle control, but similar to the surgery control that was euthanized 4 weeks earlier. So that really demonstrates the disease-modifying potential of Progenza and gave us confidence to move into our phase 1 trial. We conducted a gold standard phase 1 trial in Australia. So it was randomized, double-blind, placebo-controlled, looking at 2 different doses of Progenza. And we met our primary endpoints of safety and tolerability at both doses. But again, really encouragingly for the efficacy side, even though this was a safety trial, we were able to demonstrate significant pain-relieving effects of Progenza. So Progenza achieved a statistically significant within-group reduction in pain that was sustained right out to the 12-month timeframe, and this was achieved on both BAS and WOMAC pain scores, which are 2 validated tools for assessing pain in osteoarthritis patients. We did see some reduction in pain in the placebo patients, but not to the extent that we saw in Progenza. And consistent with what we saw in the rabbits, we saw a halting of disease progression following treatment with Progenza. So patients had an MRI at baseline and at 12 months post-treatment. And following treatment with Progenza, we were able to show a halting of disease progression. So there's a slight increase in lateral tibial cartilage volume, whereas in comparison, the placebo patients, their cartilage continued to degrade at what you would expect based on the literature, which is about 5% cartilage loss over the course of the year. So this is really encouraging results for us and gave us confidence again to move into our phase 2 trials. This slide is to give you a bit of an update on where we're at with our partnership with Kyocera in Japan. So we're continuing the successful collaboration with Kyocera. They have a number of requirements as part of the collaboration and licensing agreement. So they are responsible for the product development, manufacturing, and regulatory processes in Japan, but of course with support from the Regeneus team. As I mentioned, we will be continuing with our PMDA consultations heading in the lead up, to our phase 2 trial in Japan in 2022. In the US, we recognise that there's a big market opportunity for Progenza for knee osteoarthritis. There's quite a lot of people who have been diagnosed with osteoarthritis in the US, and this is only going to increase with the ageing population and increasing obesity, which actually is worldwide. Our upcoming milestones for Progenza in the US include completing a preclinical osteoarthritis model, various regulatory processes, including the pre-IND meeting with the FDA and our IND and RMAT designations, product manufacture, I've mentioned before, and of course, phase 2 preparations ahead of kicking off the phase 2 trial. In terms of the phase 2 trial design, of course, we could get feedback from the FDA, which would change our current thinking. But currently, we're considering a randomized double-blind placebo-controlled trial with the indication of knee osteoarthritis with endpoints that cover safety, patient-reported outcome measures of pain, stiffness and function, and of course, structural changes. So we're really looking at the disease-modifying potential of Progenza. There are no disease-modifying treatments available currently for osteoarthritis. So this is where we see Progenza having real benefits. So not only providing long-term pain relief and improvement of function for osteoarthritis patients, but having real potential to be the first disease-modifying osteoarthritis treatment available. Just like to switch gears now to take you through our Cygnus platform for pain and wound healing. As I mentioned at the introduction, our Cygnus product is a cell-free product, so it harnesses the power of the bioactive secretome only. Again, we use a very rigorous screening process for our donors. There is no genetic modification or reprogramming of the MSCs that are utilised to produce this bioactive secretome. So it's considered a low-risk product. We also have scalable production resulting in lower cost of goods. And importantly, because we're focusing on topical application with Cygnus, we have favourable shipping and storage conditions, which makes this product amenable to topical application. So I'll take you through some of the data in the upcoming slides. For the purposes of this conference, I don't think I need to go through the power of the bioactive secretome in huge amounts of detail, because many of you would be aware of all the benefits of the various components that MSCs secrete. And this includes things like reducing inflammation, reducing scarring and fibrosis, reducing infection and pain, as well as promoting things like angiogenesis and innervation, and preventing blistering and peeling. So we see that our Cygnus product could have wide-ranging beneficial effects. To hone in on some of the data we have now, we conducted a validated postoperative incisional pain model in rodents, and we were able to show that that topical administration of Cygnus just once achieved a more potent analgesic effect than morphine. This is demonstrated by this green bar here at 3 hours post-surgery, which was significantly more potent, had a significantly more potent analgesic effect than morphine at the same time point. We've also administered Cygnus to a number of human studies. So we've established safety across all these studies, including 2 safety-focused studies involving repeat insult patch testing and repeat administration of Cygnus to acne-prone patients. But really encouragingly, on the efficacy side, we have demonstrated statistically significant reductions in non-inflammatory and inflammatory lesions in acne patients, whereby 86% of these patients were considered responders. And we also saw a statistically significant reduction in the acne global severity score. We've also seen really beneficial effects from administration of Cygnus topically to age spots, including reduction in the size of those age spots and pigmentation, as well as significant improvement in the skin smoothness. So this really positions Cygnus well to be used as either a therapeutic or a cosmetic. You may have seen recently in the news that we announced a partnership with the Department of Defence to develop, with the Australian Department of Defence, to develop Cygnus for combat casualty care. So we were actually granted 300,000 Australian dollars in funding from the Department of Defence to cover 3 main areas. So this piece of work will cover optimization of the product, some additional preclinical testing in pain models, and then proceeding into a first-in-human study of Cygnus for pain. So those milestones are coming up over the next year, and we'll be excited to share the results with you. As I mentioned at the beginning, we're looking for strategic partners and collaboration opportunities with anyone for Progenza for knee osteoarthritis and ex-Japan and other indications, as well as Cygnus globally. So if you are interested in either hearing more about our product platforms or Regeneus in general, please feel free to either contact me through the partnering platform on the Cell and Gene Meeting in the Meza conference website, or please contact me on my email listed here. Thank you for your attention and enjoy the rest of your conference.